Hello everyone, and welcome to this podcast for cell biology. In this podcast, we're going to focus on carbohydrates. And throughout this podcast, I think we should be able to get this all in one podcast, but it might end up being two. I want to take a step back and talk about macromolecules. We've talked about some macromolecules, but we haven't talked about all of them yet. And I think it might be a good opportunity to kind of give a brief summary of all the macromolecules, all four of them. And then we're going to go straight into carbohydrates and talk about what is a carbohydrate. And then we want to talk about disaccharides and polysaccharides. And then we're going to close with a brief discussion on the functions of carbohydrates. So let's talk about the four macromolecules. I suspect you talked about these to some extent in Biology 111, but I think it's a good opportunity to review some of this information. And so let's think about the different kinds of macromolecules. I then want to identify what each monomer of the macromolecule is, or we might call those subunits. I want to identify the bond that characterizes each of these macromolecules. And then I want to provide an example. The first two we've already talked about, but I think it's good to put them in this table here as we give this brief background. So proteins and nucleic acids. The subunit of a protein, as we've already learned, those are amino acids. And the subunit of nucleic acids are nucleotides. Remember, there are 20 amino acids, and then we have 5 nucleotides. The bond that holds amino acids together are called peptide bonds, and then the bond that holds nucleotides together to form the nucleic acids are called phosphodiester bonds. For proteins, remember we talked about a lot of different examples. We talked about actin. We talked about several kinds of enzymes. Remember, we went through that whole list of different functions that proteins do. They play a role in structure. They play a role in signaling and communication. So a lot of different examples there. Nucleic acids, the examples for these are DNA and RNA. Okay, the next one I want to talk about here are carbohydrates. The monomers, or the subunits of carbohydrates, we call monosaccharides. And these get strung together to form more complex carbohydrates. The bond that holds monosaccharides together to form complex carbohydrates are called glycosidic bonds. Some examples, and we'll go over these later. We have glucose, maltose, cellulose, and a lot of others that we'll talk about. All right, this last one here we'll talk about in a week or two, and they're called lipids. Lipids are a macromolecule, but unlike the first three, proteins, nucleic acids, and carbohydrates, they're not polymers. And the reason they're not polymers is because they're not characterized by having repeating units of subunits. So we're going to put not applicable here. A lot of lipids, when they're being held together, are held together using an ester bond. But that's not true for all lipids. So I'll kind of put ester bond here in quotes because it's not true for all lipids. Some examples of lipids would be fatty acids and cholesterol. And again, we'll cover this in exhausting detail later on, but for now, just know what I've indicated here. Okay, as I said, we're going to focus on carbohydrates for this podcast. So let's, down here while we're on this board here, just talk about why we say carbohydrates or sugars are essential to life. We can come up with lots of reasons, but let's go ahead and list a few here. Sugars contain a great deal of chemical energy. We're going to talk about energy in a little bit more detail in the next podcast. But for now, you should know that the bonds that are found, say, in this 6-carbon sugar here, in between these bonds, as they get broken, they release a lot of energy. So chemical energy found within carbohydrates is essential. Sugars are used as building blocks. If we want, we can think about RNA and DNA. RNA and DNA require that ribose or that deoxyribose. They also serve as building blocks for other more complex molecules. They're also very important when we think about chemical evolution. We couldn't have had RNA and DNA without chemical evolution. And in particular, we should think about RNA, since we do think about RNA as being that first molecule in early life that was able to act as an enzyme and able to act as a vehicle for inheritance. Okay, so let's spend a little bit more time talking about carbohydrates. Who am I fooling? We're going to spend a lot of time talking about carbohydrates. I want to begin by thinking about some of the chemical groups that make up a carbohydrate. So all carbohydrates will contain a carbonyl group. They will all also contain a hydroxyl group. And they will also all contain hydrogens. And together, this will make up various carbohydrates. In addition to what we see here, these hydroxyl groups are attached to a carbon as well as these hydrogens. Now, we can characterize carbohydrates by the number of carbons that are present. So characterize carbohydrates by the number of carbons. So for instance, if we had 3 carbons, and that's really the minimum, we would say that that carbohydrate is a triose. Tri for 3, O's for the carbohydrate. We'll also talk about 5 carbons. These will be pentoses. And then 6 carbons would be hexoses. Hex for 6, oses for sugar. Up here for pentoses was pent for 5, and then oses for sugars. These are 3 very common carbohydrates that we'll find within a cell. Now, interestingly, you could have 2 trioses with the same atoms but are different sugars. This is also true for the pentoses as well as the hexoses. I'm going to start by talking about 2 examples for trioses, and then I'm going to talk about 2 examples of the hexoses. So let's begin by looking at aldoses and a ketose. Both are 3 carbons, and you could write both of them like this: 3 carbons, 6 hydrogens, and 3 oxygens. But they're different. So let's talk about how they're different. Here's our carbonyl group. Here's our carbon, the second carbon here with an hydroxyl group on it, and then a hydrogen. And then we have a carbon, the hydroxyl group, a hydrogen, and another hydrogen. Remember, the important thing about carbons is that they can form 4 different bonds. So that's what we're seeing here. And we always number sugars typically like so, 1, 2, 3, with the 1 being near the carbonyl. So let's draw the ketose. We have our carbon and our hydroxyl group with a hydrogen. We then have our carbonyl here, and a carbon with a hydroxyl and the hydrogens. So you can see that the only difference here is the positioning of the carbonyl, with the aldose having the carbonyl up here at the first position, and the ketoses having the carbonyl at the second position. So this is the first way that carbohydrates with the same atoms can be different carbohydrates. And so that first way is placement of the carbonyl. Now this is important. For instance, aldoses will be used as a building block for a set of molecules, and ketoses will be used as a building block for a set of different molecules. And it's all based upon the positioning of that carbonyl. Now, the second example of how you could have 2 carbohydrates with the same atoms in it, yet they are different sugars, has to do with spatial arrangement of the atoms. This is a much more subtle difference. And to look at this, I want to look at a 6-carbon sugar. And both of these 6-carbon sugars are derived from an aldose. The first one is a glucose, and then the second one is a galactose. Both of these would be considered aldoses because they are derived from that 3-carbon aldose sugar. I'm going to draw the glucose first. Okay, now I'm going to draw the galactose. And when I'm done drawing it, I want you to look at the 2 and tell me if you can tell the difference. Again, it's a pretty subtle difference. Okay, so as you're looking at them, there's only one very small difference. And that very small difference is found at that 4th carbon. So we have 1, 2, 3, 4, 5, and 6. 4, 5, 6. And the difference, as I said before, is very subtle. And it's just that spatial arrangement difference at this fourth carbon of the hydroxyl group and the hydrogen. You can see here the hydroxyl group is on top, here's the hydrogen's on the bottom. Hydrogen on top here, hydroxyl on the bottom. Now, on a test or a quiz, I'm not going to show you these two structures, and you have to tell me if it's glucose or galactose. We'll save that for when you take organic chemistry or biochemistry. But what I do want you to know is that two sugars that have the same number of atoms and same types of atoms can be different sugars because of either the placement of the carbonyl or the spatial arrangements of the atoms. And that can make all the difference in how these are used, because remember we said those aldoses and ketoses can be used as different building blocks. Here what happens is that the glucose is the major source of chemical energy in the cell, because we have enzymes in our cells that can break down glucose into energy. And we'll cover that in a lot more detail later in the semester. Galactose cannot be used by all cell types. And so for it to be used, it often has to be converted back into glucose. Now I want to talk a little bit about how sugar naturally appears in our cells. Pentosis and hexosis, and anything higher than that, will spontaneously form a ring. And the way it does that is that this hydroxyl group, particularly the oxygen here, on this fifth carbon, will form a bond with the carbon at the first position. And that's going to wrap this all the way around. And so typically we will write sugars in a cell looking something like this. And for glucose, we call what we see here as the alpha form. So this is alpha glucose. It also exists as beta glucose. And the only difference between alpha glucose and beta glucose is the orientation of the hydrogen and the oxygen. So for beta glucose, it would look like this. Within a cell, as this forms this ring, you'll have equal amounts of alpha and beta, but it turns out that the beta is more stable, and so you'll see more of the beta glucose available within a cell, because alpha glucose is less stable. Now, I'm not going to make you draw this on a quiz or on an exam, but you should recognize this as being glucose. And you should also know that it exists as an alpha form and a beta form, and that the beta form is the more stable form. Now, glucose and galactose that we talked about on the last slide are called monosaccharides because they exist as a single ring. But sugars can exist in structures containing multiple monosaccharides, sometimes 2, sometimes 3, and sometimes hundreds. So let's talk about the more advanced structures and move beyond the monosaccharide. So let's begin by talking about disaccharides. And then we'll take that discussion into polysaccharides. Okay, so let's create our first disaccharide. And what we're going to do is we're going to take two glucose molecules, and I'm going to draw a simplified version of the glucose molecule that we saw on the last slide, where we have our oxygen here, and then we have our hydroxyl group and our hydrogen. So this is glucose. And we're going to add that to another molecule of glucose. So remember, when we build a molecule like this, a larger molecule, we call this a condensation reaction. Condensation reactions will take these two monomers, put them together, and it's going to release water. So let's look at what this disaccharide looks like. And the resulting disaccharide here is called maltose. So you should know that one glucose plus another glucose equals maltose, which is a disaccharide. Maltose is found in a lot of things we consume. It's found in cereal, pasta, beer, and other food products. When we consume maltose, our body cannot use maltose by itself. It has to be broken back into two monosaccharides of glucose. And it does that using a hydrolysis reaction, where it will use a water molecule to create 2 glucose monosaccharides, which then can now be metabolized in the cell to create energy. Another disaccharide I'd like you to know is lactose. And I'm not going to draw this one out, I'm just going to draw the word. So lactose, which is the sugar that's found in milk. And you make lactose also by condensation reaction, but by using a glucose plus a galactose. These 2 monosaccharides together will create lactose, also releasing water, because it is also a condensation reaction. Now we're going to jump from these disaccharides straight into polysaccharides. Polysaccharides typically contain hundreds of monosaccharides. So let's talk about some of these. Alright, so there are 5 different polysaccharides I want you to know. And I'm going to list them here in just a moment. I want you to not only know their names, I want you to know a little bit about their chemical structure. And then I want you to know the organism that we find them in. Sometimes we find these only in animals, sometimes we find them only in plants or bacteria. So I want you to know that. And something about their function. Let's start with the starches. We could describe these chemically as a linear array of glucose molecules with few branches, if any. And so what I mean by that is, if this is my glucose molecules, they're going to be stretched like this and connected to each other in a fairly linear manner. Every now and then you'll see a branch coming off, something like that, but not often. These are found in plants. You think of starchy food like potatoes, that's what we're talking about. So it's found in plants, and plants use them as an energy store. When we eat these, we have to break down this linear array of glucoses so we can use those glucoses. All right, the next one I want to talk about is glycogen. I'm going to keep this also in orange because starches and glycogens share a lot of features. They're also a linear array of glucose molecules, but they are highly branched. These are found in animals. And just like plants use starch as an energy store, animals use glycogen as an energy store. And I just realized I misspelled glycogen, left out the O. In animals, we typically will find these in muscle tissue and the liver. That's where we store a lot of our glucose so we can use it later. Okay, the next 3, I don't know if I'll get them all on this board. I may have to erase something here, but the next 3 play a larger role in structure and support. The first one I want to talk about is cellulose. Like starches and glycogens, they also make this linear array of glucose, but unlike these 2, the starches and the glycogen, these arrays will stack up on each other in a parallel manner. So they might look something like this, where you have these repeating glucose molecules, and they'll have these different strings of them, so to speak, but they're connected with each other, which is why they provide a lot more structure, a lot more support than you might expect to see in starches or glycogens. And since starches and glycogens you need to break down pretty quickly, the last thing you want to do is create more bonds that are holding them together. So we typically will find celluloses in plants and many algae. They're part of that cell wall that provides that support. So when you eat a celery stick, what you're eating is primarily cellulose. When you eat a salad, that tough structure on the outside of it, that is cellulose. All right, next I want to talk about chitin. Chitin is like cellulose in that they form that linear array, but it's not of glucose. This is where it's different. The sugar it uses is called N-acetylglucosamine. And it'll look very similar to what we see here, except instead of glucoses, it'll be these N-acetylglucosamines, which we often just call NAGs. I want to make sure I keep these separate. I'm kind of starting to merge some things here. So chitin we find, just like with cellulose, I didn't say it here, let me write structure here. They're important in structure in insects and fungi. In insects and fungi, chitin is typically found in fungi, and in fungi it's part of the cell wall, or insects, and in insects it's part of the exoskeleton. Within the cell wall or the exoskeleton, they help provide protection for the organism, structure, and support. Okay, for this last one, I'm running out of little room on the bottom, so I'm going to write it up on top here. I erased the first two here, but I want to shortchange this one because this one's a very important one. They're all important, but this is a very cool one. So these are called peptidoglycans. Like chitin, they will also use the N-acetylglucosamine, but instead of repeating units of N-acetylglucosamine, they will bind with N-acetylmuramic acid. You can just know them as NAG and NAM. Like cellulose and chitin, they will stack up upon each other to provide that extra support. And just like plant cells and insects and yeast and algae, there's one other organism that we haven't talked about that also has a cell wall, and these would be bacteria. Bacteria use the peptidoglycans in their cell walls. I think it's appropriate for us to spend a little bit of time talking about the peptidoglycan structure here. And the reason is that it's a key feature that microbiologists use to distinguish between gram-negative and gram-positive bacteria. What microbiologists will do is they will stain bacteria with a dye. The dye is called crystal violet. I'm not going to ask you that on a test, but I'm going to write it down here just so you know in case you're curious. Gram-positive and gram-negative, they will each have peptidoglycan in their cell walls, but gram-negatives has a thin peptidoglycan wall, and gram-positive have a thick peptidoglycan wall. For that reason, gram-negative bacteria stain poorly. You'll get a little bit of a stain, but it's not quite as strong as you see with the gram-positive. Okay, the last thing I want to talk about is treatment. If you have a bacterial infection that's gram-negative or one that is gram-positive, which one is easier to treat? Turns out for gram-negative bacteria, these are more difficult to treat. Now, it should be pointed out that most bacteria that we come in contact with on a daily basis, or the billions of bacteria that live within our own body, are not harmful at all. In fact, they're useful. We need them. So sometimes we have a negative connotation of bacteria that they're bad for you, but most of them are good. But there are a few that do make us sick. And the ones that make us sick that are gram-negative are more difficult to treat. For instance, some strains of E. coli would fall into this, but of course not all. Gram-positive bacteria are easier to treat. Some examples of gram-positive bacteria would be like streptococcus. They'll make you sick, but antibiotics are usually pretty effective at treating them. Okay, that's all I want you to know about these 5 different polysaccharides. Now, the last thing I want to talk about are carbohydrate functions. And I want to primarily talk about 4 different functions. And we've talked about some of these already. One of the functions we talked about before was that they provide a substrate, or earlier we call them building blocks, for making more complex molecules. And remember, we talked about how the ribose in RNA and the deoxyribose in DNA are examples of how carbohydrates serve as these building blocks. They provide structural support. And we've talked about that already when we talked about cellulose, chitin, and the peptidoglycans. Now, this next one is something we haven't talked about yet, so let's spend some time with it. And we're going to call this cell identity. You may not know what blood type you are, but you're probably aware that blood can either be blood type A, it can be blood type B, it can be blood type A, B, it can be blood type O without either of them. So what makes these blood types A, B, or O? It all has to do with the carbohydrates on the outside of the red blood cell's membrane. Let me draw the membrane in a little bit more detail. And we'll talk about membranes later in a week or so. But these membranes can sometimes have proteins attached to them or going through them. And these proteins may have various carbohydrates attached to them. These lipids here may also have carbohydrates attached to them. I should make this look more lipidy. When they're attached to a protein, we call them glycoproteins. When they're attached to a lipid, we call them glycolipids. So what do? Well, they help protect against the immune system. They play a role in identifying this cell as being part of oneself, so our immune system doesn't attack it. That's why if I'm blood type A, my immune system won't attack those blood types. And if you give me somebody else's blood that is also blood type A, my immune system won't attack it. But if you give me blood type B, my immune system is going to go after it. So they help protect. They also play roles in signaling pathways, and we'll talk about those later in the semester. And they're very important in development. And what I mean by that is if you have a sperm cell from one species and an egg cell from another species, and then there happens to be another egg cell but from a different species, how does this sperm know to fertilize the egg of the same species? It largely does it because of glycoproteins on the membrane of the sperm and on the membrane of the oocyte, the egg. And so it knows which egg to fertilize. And it knows not to fertilize this one down here. Okay, the last one I want to talk about is energy. Carbohydrates are very important in energy. And we're going to spend a lot of time talking about carbohydrates and energy as we move throughout the semester, but I want to introduce some things right now. Glucose is a very common sugar that cells will use to make energy. And often that form of energy comes as ATP. In making this energy in the ATP, it'll also make some other molecules that we'll talk about when we start talking about cellular respiration, but we'll wait for now. But why does glucose have so much energy? And we briefly talked about this earlier, but it's because of these chemical bonds here. Each bond between this carbon and this carbon has a very specific amount of what we call potential energy. And we'll talk about potential energy in the next class, but for right now, you should know that all I'm saying there is that when that bond is broken, it's going to release a lot of energy. But before it's broken, all we can say is it has the potential to have a lot of energy. But because we have all these bonds here, and they're not super strong bonds, it has a great deal of potential of generating energy. So why is it then different structures have more or less potential energy? We can think about CO2, we can think about our good friend glucose here, and then fatty acids. We know that CO2 has very little energy, potential energy in it. It's really hard to get any energy from CO2. So we're going to say low energy here. Sometimes when I do energy, I put an E with a kind of a lightning bolt there. So if you ever see me write I mean energy. So low energy for CO2. We're going to say medium energy for glucose and high energy for fatty acids. And the reason for that is because the covalent bonds in the CO2 molecule are held together much tighter. Glucose has more energy potential than CO2 because those bonds, those covalent bonds, are not quite as tightly held together. And then fatty acids is the other extreme where there's a lot more potential energy in here because it's not going to cost as much energy to break those bonds, but it's going to release the same amount of energy. So they have the weakest covalent interactions. Now, not all the energy that enters our cells is glucose. Remember, sometimes it has to be converted to glucose. And if you remember, we talked about starches. So if you eat some starchy food, a potato, a piece of bread, that starch has to be converted into glucose. And it's converted into glucose because of an enzyme called amylase. Amylase is found in our salivary glands. It doesn't need to be found anywhere else in our body because we don't make starch. This is just in plants. And when we eat plants, we want to be able to get that glucose. And so our salivary glands release amylase, which breaks it down into glucose. Now we have another source of glucose in animals called glycogen that we talked about earlier. It also has to be broken down to glucose. We can't just use glycogen. It's got to be broken down. And it's broken down using an enzyme called phosphorylase. Phosphorylase is only going to be found in cells where glycogen is present. And so we talked about that earlier, but let me remind you, muscles and liver. This is where most of our glycogen is stored. And so phosphorylase enzyme is going to be present in muscle tissue and liver tissue so that it can break down glycogen to be used as glucose. Wouldn't make a lot of sense to have a lot of phosphorylase in our saliva. It makes more sense to have it in our muscle or our liver. All right, that's all I have for this podcast. If you have any questions at all, please let me know. If not, I'll see you in class. Bye.